it's nice that we can all, come together as a global community as we have before. I believe we did not have an international Catchathon last year in 2020, so I'm really excited that that we had the wherewithal to do it again this time. And I have to thank my teammates and Agla Ramanauskite in particular for suggesting that we do this and that we do it quickly for Citizen Science Month. And so we're at the very end of April, which happens to be Citizen Science Month. So it's an opportune time to do this. And it looks like we are being joined by a lot of our old catching teams. So we have some of our original ones. We have folks from Nigeria and from Uganda that have confirmed their participation. And I expect when we start looking at the team activity, we're going to see a lot more of some of our old favorite teams who have been with us since early on. And I just want to take a moment to wish Mike Caprero a happy wedding anniversary, and thank you for taking time out of your day to join us for this, and also to acknowledge that Mike has been catching stalls since the very first day that we launched this project, I believe, from the very beginning. And so it's an honor to have you here, and you're still catching very actively on a daily basis, and this is 4.5 years later. And I think you just had a major milestone in that you almost turned the odometer, so to speak. So stall catchers, we would, in the database, you have to say, okay, the score will never be higher than this, so we don't need to take up more space than this. But Mike has proved us wrong, and we actually needed to allocate more memory just to hold Mike's score. So thanks so much for all your dedicated catching and research analysis that you've contributed, and to all catchers out there who have participated through these years and who continue to participate, and some of the new folks who have joined us today. So the theme of our kickoff today is Meet the Scientists, which is why we have a bunch of scientists here on the chat, and so I'd like to before we get started with the biomedical science of stall catchers, I'm just going to say a quick word about a special aspect of this event, which is that we have a new kind of catcher participating, which is an AI-based catcher, a bot that's been wrapped in what we call an agent wrapper, and that's participating as a player alongside other catchers. We did a quick test on the live system before the Catchathon went live, and things were going smoothly. I just heard that something came up, and I don't know. So our bot's name is GAIA. It was created by Laura Onak from Romania, who was one of the winners of our Clogloss competition for machine learning. There were over 1,000 participants in that competition. So to be one of the winners of that competition is a big deal. And we're grateful that Laura's joined our team now to continue to do work in this area of automating stall catchers analysis. So Laura named her bot GAIA, and GAIA is a very competent stall catcher. At the moment, maybe having some cold feet on the day of the Catchathon, but we'll check back in with Laura in a bit on that. Our main goal for the human computation research related to this is to figure out whether or not, if you take an AI system and you have it play alongside human players, and then you take answers from human players and AI players and combine them together, do we get at least as good or possibly a better answer through that rare combination? Our wisdom of crowd methods relies on diversity of players and diversity of how they see the vessels and come up with their answers. And we think that the AI bot might be sufficiently diverse to add value to this process. And of course, AI bots don't need to sleep, so they can be running all the time, and maybe we can get this analysis done even faster. So with that said, why don't we go around, and I'd like to begin with Diane Bovenkamp, who joins us from Bright Focus Foundation. Bright Focus was, of course, the organization that gave stall catchers its wings. So Diane, I wonder if you could introduce yourself and just say, and introduce Bright Focus. And here we are 4.5 years out. And maybe you have a few words about what this project has, what significance it's had for Bright Focus.
Well, yeah, I think it's been absolutely phenomenal partnership with so many people. There's so many moving parts and scientists involved with this, but I'm Diane Bovenkamp, Vice President, Scientific Affairs at Bright Focus. We are have funded over the past 48 years that we've been in existence. We're a nonprofit funder for finding cures for Alzheimer's, glaucoma, and macular degeneration. And so, yeah, I guess it has been is 4.5 years now that Dr. Michelucci, Pietro, brought this crazy little idea to us. And so we're really honored and proud that we were able to help fund the creation of Eyes on All stall catchers, as well as we actually funded Dr. Chris Schaefer and all associated scientists with both of those grants to then use stall catchers and for all of you to actually use this game to play and analyze data. So so play a game, help advance research. That's kind of been the motto. And it's been it's beyond phenomenally successful, beyond our wildest dreams that we've been able to create it. You had PBS special. You've had so many people around the world participate. And for me, you know, the most exciting scientifically was the Science Reports publication in 2020 that all of you stall catchers had an opportunity to have your names associated, acknowledged with a scientific paper. I think, Pietro, that was the first one that was out so far, or one of them anyways, for the Alzheimer. That was the second one where people's but yeah, so it shows that everything that you're doing here today is helping to advance science, and that's a really high impact publication. So we're extremely pleased.
Yeah, I appreciate all those kind and supportive words, Diane, and I definitely want to give Chris Schaefer credit here, because I think Chris, from the very beginning of the project, so Chris and Nozomi lead the lab that's been doing the biomedical work for anyone meeting them for the first time. And I think since the beginning, Chris has really appreciated and supported the citizen scientist aspect of what we're doing. It's always been, I think, much more, you know, from what you've told me, Chris, much more than just getting data analyzed. It's been about educating people and doing this all together and recognizing the accomplishments. And I think you fought for, I mean, you tell me, but I think you fought with, you know, advocated, I should say, to have everyone you know, mentioned in these papers as much as possible. So why don't we, thanks Diane, and why don't we switch over to Chris? If you could just do a quick introduction and tell us a little about what stall catchers over 4.5 years has done for your research.
Yeah, sure. So I'm Chris Schaefer, faculty member in the Mining School of Biomedical Engineering. I run the lab together with Nozomi Nishimura that's focused on trying to understand the causes and the consequences of brain blood flow reductions in Alzheimer's disease. And I think as this community appreciates in a very visceral sense, those blood flow reductions, at least in mice, seem to be caused by these stalled capillaries. And it's just so time consuming to do this data analysis that when we first discovered this phenomenon, it was sort of it was spooky because we kind of laid out some of the key questions we felt like we needed to answer in order to know whether this is really impactful in humans, or how we might modulate it, or who would be most likely to have these stalls. And when we looked at those questions and how long it would take to analyze the data we need, it just didn't seem possible. And stall catchers has really fixed that problem. There's already now 3 reports on data that the primary analysis of whether capillaries were flowing or not was done by stall catchers. So one report was actually a suggestion that came from the stall catchers community, asking whether or not exercise could be a factor that mitigated capillary stalls. And it's a good hypothesis. Exercise has very diffuse and always very diffuse, always positive, and complicated effects on physiology. So, including there's evidence of decreased vascular inflammation. So, so we launched a study initiated by ideas that came from the stall catchers community. Unfortunately, while it is very clear that exercise both helps with Alzheimer's symptoms and with all kinds of aspects of your physiology, it does not seem to eliminate this capillary stalling phenomenon. So in some sense, that's a fantastically important thing to discover, because it means just exercising and being in good shape isn't enough to defeat this problem. So that paper was published last year in PLOS One. We also had another big paper that Diane mentioned that was published in Scientific Reports. That paper focused more on the question, if we had a therapy that would interfere with this capillary stalling effect, who would you give it to? And if you wait until you're well into developing Alzheimer's disease, there's a sense that maybe there was a missed opportunity there. And so our idea was, well, maybe some of the risk factors, the things that are, like having high blood pressure or having high cholesterol, these things that are linked to increased risk for developing and increased severity of Alzheimer's disease. Maybe some of those things are acting by increasing the incidence of capillary stalling. And so the first thing we tried was just obesity, because it is such an important risk factor for dementia. But as you know from that paper, it was another negative result, where it turns out that obese mice mice being obese doesn't seem to synergistically make the capillary stalls worse along with Alzheimer's disease, suggesting that the way that obesity impacts and worsens Alzheimer's disease is through an alternative mechanism. And then there's a third report that we're very excited about. It's in peer review right now, but we were so excited about the results that we've put it up on a preprint server, bioRxiv. And this is a manuscript that shows that a compound that is a commercially available and approved drug does eliminate capillary stalls very rapidly in the Alzheimer's disease mice. This is a drug that blocks the signaling of a molecule called vascular endothelial growth factor, or VEGF. And these kinds of drugs are often are widely used for both treatment of macular degeneration. Diane, as you probably know from the Bright Focus on the Bright Focus Foundation's focus on vision loss. And it's also given for some kinds of cancers. And in both of those cases, it tends to prevent growth of blood vessels. But for mechanistic reasons, we are still investigating. In fact, we're writing a grant right now to try to understand why it is that this anti-VEGF is working as well as it is. But nonetheless, this is a report of a drug that there's versions of this that are on the market and prescribed to patients. And we've shown in mice that it eliminates capillary stalls and improves brain blood flow. And that was, again, all of the stall analysis there was done by stall catchers, and stall catchers contributors are listed as authors on that manuscript. So this is, three really important, findings that built on the initial work that we had done in the lab. That's all enabled by the work of stall catchers.
So if I could just add, just jump in there with one thing that you did mention about the anti-VEGF drugs and this crossover, I think it's, this is where, with science, you test a hypothesis and it either works or it doesn't. But if it doesn't work, the initial hypothesis, it doesn't mean it was all a waste, right? With science, then you can just use that information to retool and come up with another question. And sometimes it takes you in a really innovative idea that might just be the next best idea to find a cure. And so, yeah, and there's lessons learned from other diseases, right? This crossover. So I'm sure that Chris will be going to look at macular degeneration research, might even bring in a macular degeneration researcher to try and see, because there's blood vessels involved in both diseases. And so great.
Yeah.
And so this is where the catchers here are perhaps helping to start the basis of something revolutionary.
Yeah, and it's definitely the case that we're often wrong in science, and science can be a very humbling endeavor. So to be honest, those three findings that I just described, I was 100% wrong on my initial instincts for those three reports. So my initial thinking, and not just what popped into my head, but based on my understanding of the literature and the mechanisms involved, I was confident that, hopeful that exercise would decrease capillary stalls and increase blood flow. I was confident that obesity was going to dramatically increase capillary stalls, and I was hopeful that eliminating them would get rid of the way that obesity makes Alzheimer conditions worse. And I was strongly opposed to testing anti-VEGF therapies in my own lab, because there was a relatively junior member of the lab. He was an undergraduate at the time, mentored by at that time a relatively junior postdoc in the lab, and they came up with this idea, and I just didn't buy it. I felt like the science wasn't the science didn't fully support it. There were parts of the mechanism that weren't there, but I've learned, you shouldn't only trust yourself. And so after I made clear why I didn't think that experiment would work, I got out of the way, and Mohammed and Oliver disagreed, and they moved forward with the experiment, and they were right about the effects of anti-VEGF. Now, I do want to clarify, the mechanism that they thought by which it worked through isn't right. So I was right about that, but it did turn out to work. So anyway, this is a very, and this is not an unusual way that science works. And I don't think it means that, I'm dumb because my instincts weren't right on these studies. This is how science should work. We're going, past the edges of the map in some sense. Nobody knows what the right answer is. And so we try to design experiments and ways of testing ideas. And we're wrong more than we're right. The key thing is to be very honest with what is the data telling you? What can you really know? What do you suppose? What do you hypothesize based on that data? And really, being your own worst critic in trying to uncover what the data is really showing. Once you've sort of taken that step beyond the edges of the map. So in the case of these three papers, yeah, I was 100% wrong when we were initiating the studies, and 100% proud of the findings that we ultimately came to with the help of stall catchers.
Yeah, Chris, I think it's a real testament to your leadership and your character and your humility and the ethos that you set in the lab there. And I realize that it may not be just a philosophical stance or ethical stance, but that you actually end up getting the most value out of what you do by, I think, approaching science with humility, which I think is part of what you're saying. So I think these are really encouraging results, and I think one of the in human computation, we often try to kind of measure the amount of information that we're getting from different processes, and when you describe having entered into these research questions, having a certain bias or expectation, and then being surprised, we often think of surprise as sort of the the biggest amount of information you can get. When something happens that's totally unexpected and not what you predicted, it means that you now have information you didn't have before that's going to compel you to revise and improve your model. If everything happened the way you expected to, it's not really that much more information. It's just kind of like, okay, yeah, we were right about that. So, what's the next thing? So I think surprise and disruption can be instrumental for the advancement of science. And, as we know, historically has compelled some huge paradigm shifts in our thinking about just about everything. Just really grateful for this ongoing collaboration that we have with you and Nozomi and the great folks in your lab, and all the volunteer catchers, and the folks like Diane and Bright Focus who have enabled this work. Let's see, we're about halfway through. I just in a quick spot of good news, if you're pro AI, it sounds like GAIA, the new bot, has recovered from its stage fright. I'm still not clear on the pronouns that we're supposed to use for GAIA yet, but I'm going to say it for now until someone corrects that. But it sounds like GAIA is underway, and who knows, maybe we'll show up on the leaderboard. It is a very computational intensive process. So even though we stood up a pretty powerful server with GPUs, GAIA, I think, is still annotating 1 or 2 videos per minute, which is slower than I think a typical human plays the game. But on the other hand, GAIA is going to be running all night long, and slow and steady wins the race, right? So we'll see what happens. And so now I just like to, since we're still kind of on the biomedical topic, is just Leanne, I wonder if you could just quickly unmute and introduce yourself and your role in the lab, and tell us a little bit about this APOE data set, which I think comes from your own research, which is now becoming the largest data set ever analyzed in stall catchers. And tell us why that's important to you, and why we're studying that and analyzing that.
Okay, so hi, everybody. I'm really excited to be here. So this is my first, I guess, Catchathon, and I suspect I'll be here for another few years to come. So I'm a new postdoc that joined post-pandemic in the lab, so June 2020. And yeah, so I'm working with a PhD student, Nicole, on this so I say ApoE, so apolipoprotein E mouse model. So basically we've strayed a little bit away from these, I guess, traditional Alzheimer's disease mouse models that focus a bit more on a particular specific protein that definitely does have a role in the disease, but we're looking more at these risk factors that have been strongly associated with later risk of developing dementia. So this is actually the strongest, so ApoE, if you have a certain form of it, so there's 3 different variants. If you have the E4 copy of this gene, you're actually at a higher risk of later developing dementia, and it's the strongest genetic risk factor for this late onset version of Alzheimer's disease. And if you have 2 copies, you're at an even higher risk of developing dementia. And it's actually quite common in the population. So about 15 to 20% of individuals have this copy of it. The most common form is the E3. So we're comparing so that's kind of our control group. We have these E3 mice versus E4. And we're just starting to see whether or not these mice actually do have a higher incidence of these capillary stalls. So we've looked in younger mice, and we've also looked in older mice to see if age had a contributing factor. And this data is kind of just coming in slowly. So we're definitely relying on stall catchers as well to help confirm what we think we've seen now. And now more recently, we also added an important cardiovascular risk factor on top of this. So we've added hypertension to these mice, and it seems pretty promising so far that when we added hypertension to these mice that have the E4 copy of ApoE, that they have this increased incidence of stalls. And this also seemed to correlate with memory performance. So their memory also seemed to have shifted downward. And this, we had also added hypertension to our controls, and they did not have the same impairment. So we're excited to see if stall catchers can confirm this. We're also interested in looking at whether or not it's a similar mechanism that we've seen stalling occurring in this other Alzheimer's mouse model. It looks like it might be happening through a different pathway, but there might be somewhere that they converge. And we're interested now in seeing how different treatments might be able to mitigate this. And we'll see if it it overlaps a bit with what we've already seen, or if there's something new happening. So yeah, that's.
Well, I think that's a great summary. Thanks a lot, Leanne. And I think one of the things that interests me about what you described is that it recognizes that there's often a confluence of factors with disease. So, you know, someone might have a genetic predisposition to a disease, but then their lifestyle doesn't introduce, you know, they have a healthy diet and they exercise, but if you can create the perfect storm, you know, as I think you've intentionally done in some of these treatment group or study groups, then you start to see effects from the combinations of those things. So it just, you know, makes me wonder, you know, what's the take-home lesson for folks, you know, when you hear about studies that say, you know, a high-fat diet leads to this, or 15% of people are genetically predisposed to this. You know, does that mean it's your destiny, or does it kind of suggest some kind of control over your outcome?
It's an excellent question. So yeah, there's definitely. Let's restart. So basically, for Alzheimer's disease, there's an early onset form of it, and there's a late onset form. So the early onset is really only representative of 1 to 2% of the population, I would say. Some people say as high as 5%, where if you do have these certain genes, that you're essentially guaranteed to develop the disease. But really, the majority of the population, it seems to be a combination of risk factors, most of which I would say you do have a certain level of control over through diet and exercise, and even just treating these cardiovascular risk factors. So there is a lot of evidence that if you treat hypertension with certain medications, or if you treat diabetes with other medication, that you can actually decrease your increased risk of developing dementia later in life. So there are some genes like this ApoE gene and a few others that are associated with the late onset form, but just because you have them doesn't by no means guarantees that you will develop dementia later in life. So
That's
yeah.
That sounds nicely reassuring, I think, for anyone who yeah, wants to know whether or not they can impact their future health. Thanks a lot for joining and being part of this, you know, your first Catchathon here, and looking forward to working with you over the next few years. Why don't we switch now over to a couple of the folks. We have Nancy and Mike Lamont here, the other Mike, and so I'd just like to ask you to briefly introduce yourselves, maybe starting with Nancy and then going to Mike. And one of the ways in which we appreciate, I know you make many contributions to the lab, including research, Nancy, and I know, Mike, you're involved in a lot of stuff there. But lately, we've been relying on both of you to do something really important, which is to take the raw data from the research that Leanne is producing and get it into a form and insert it into stall catchers so we can actually analyze it there. And I think this is often an unintentionally veiled part of the process, and I'd like to unveil it so the rest of the community can see how much work is going on in order to get the data into stall catchers in the first place, and even maybe talk a little about how we're hoping to improve that. So Nancy, would you like to introduce yourself first, and then Mike, and then we can talk about that?
Yeah. Hi, everyone. I'm Nancy. I am a PhD student in the lab with Chris. I work a lot on Alzheimer's disease. My main project is focusing on understanding the contribution of oxidative stress to capillary stalling and blood flow in this mouse models of Alzheimer's disease. As Pietro mentioned, when we take our data from the microscope, sometimes we just have to process it before we send it to stall catchers. And part of that processing consists in kind of masking away some of the vessels that we don't need to be analyzed. For example, large vessels, those are the ones that don't get stalled. So we only want to have the capillaries, right? Because those are the tiniest vessels that get obstructed with a white blood cell. And so we just have to mask all of those areas that we don't want people to score because we don't really need that data. And so all this, we do it manually. And it obviously increases the work when we take more and more and more data. We have to go through each single one of our videos and mask all those regions that we don't want to analyze, right, or send to stall catchers. So yeah, that's one of the things that takes away a lot of work. And the second thing is, after we have done all the masking, Mike is the one who takes care of this next step, which is basically segmentating all those vessels, forming like a skeleton of the whole stack, so that then we can extract a total number of vessels, and then we can get a stall rate, or the amount of stalls over the total number of vessels. And this also has a lot of challenges, so I'm gonna let Mike talk about them, but yeah, that's the main point.
Okay, that's perfect. Thanks for describing, yeah, that masking part of the process. So yeah, Mike, if you could introduce yourself and say what happens next, that would be great.
Thank you. So I'm Mike Lamont, joined Schaefer Nishimura Lab quite a few years ago now, I guess. And one of the things that I took over was this stall catchers processing from Mohammed Haft, who had done a lot of the initial work with you on this, including the what he calls deep VES, which is the part that takes the entire volume that the biologists image and sections it between vessel and non-vessel. And of course, one of the questions that we want to ask is, if we consider a vessel is a branch between, or the vessel between 2 points as say a segment, we want to know how many of those segments have stalls in them. So the tricky part is to take that volume of data and from that just to draw the entire map of the vessel structure inside the volume. And it's a pretty tricky thing to do. And Mohammed had initially used machine learning to do that initial segmentation, which then just sort of says masks what's vessel, what's not vessel, and then post-processing, and from there, post-processing is then to skeletonize it or thin it down to get down to just a single pixel long map of the vessels inside that volume. And some of the standard processes for that, it gets confused when it comes to things like large vessels. I usually think of it as sort of like a hoodoo. If you look at desert regions with hoodoos, and you have erosion from surface down, you have little things that will sort of stick, and then things will erode below that. So you take something that's a large vessel, it doesn't always erode down to a single pixel. It'll erode, things will get caught, and then it'll end up being sort of like this line with lots of hairy stuff sticking out of it. And it's one of the reasons we need to get rid of those larger vessels for masking during the masking process before we do the skeletonization. It's worked sometimes it hasn't. One of the things we're trying to do is a fairly intensive process for the biologists. I don't do any of the biology side. I'm a physicist, so I just do the imaging side and the equipment side and the data side. But it's fairly involved for them to do that. So we're working on some ideas on, in fact, I've got a window over here on the side here, working on some automated ideas on how to computationally mask out the larger vessels. But I guess most of the stuff that I work on in the lab is trying to improve the quality of the stuff that we send off to stall catchers. And the feedback that we get from stall catchers is quite beneficial. We sent in recently some 3-photon data, for instance, and I was really curious to see what did the stall catchers, what do the catchers think of that 3-photon versus the 2-photon data, and getting feedback on that sort of thing is really good on giving us an idea of, I can look at an image, 2 images, and I can think, well, this one looks better than that one. But in practicality, it's really, really useful to have that feedback from the catchers on on what they think in terms of what's easier for them to analyze, and that helps us in the future decide which direction we want to take our research.
Yeah, and I think it was actually, so it was the mic to mic connection here in that I think when we took your question about the quality of the 3-photon data and we brought it to the chat, I think Mike Caprero was actually one of the folks who said, yeah, this actually looks pretty good, you know, compared to some of the other ones, if I'm remembering right. Do you remember that, Mike? Or you're muted still, I think, there.
I remember commenting about a variety of things. I can't remember that particular one specifically.
Yeah.
I do remember another data set that I thought was an Andy Warhol data set.
Yeah. And that's part of our processing as well, that sometimes works and sometimes doesn't. So it's always a trade-off. Do we try to make the data set look nicer, that has more processing, that ends up with, yeah, the Andy Warhol effect. So.
Yeah. And I think it's interesting in that, you know, there's this kind of ongoing sort of, we have different tools at our disposal. So we want to do masking, and we've been taking automated approach or investigating automated approaches to masking, where we use, you know, some version of machine learning or just fancy computer vision algorithms to try to identify these large vessels and then mask them. But in the meantime, you know, we're still trying to bring data into stall catchers, and the masking can sometimes be a bottleneck to that process. So we've also explored, it looks like Nancy might have had to cut out here, but Nancy and I were talking with a graduate student at Dartmouth who was interested in doing a class project related to citizen science and human computation for a citizen science class he was taking there. And so we said, you know, what if we wanted to make this a new stall catchers task, to actually bring the masking part of the process onto the platform and see if people want to try their hand at that. How could we get a bunch of different masks, for example, and combine them? So if my mask is sort of roughly good and somebody else's is roughly good, how do we bring those masks together? Is it more important to block out everything that every person thinks should be blocked out, or just take the intersection and only barely block out what everybody agrees on should be blocked out? That kind of question about consensus. So the student, I just got an email from him last night, and he sent me a YouTube video. And I don't have it at my fingertips, or I would show you, but it's basically, it's like a Homer Simpson with a donut, and the video is about masking the donut. And he came up with kind of an approach to this that we could potentially test out in stall catchers if we wanted a stopgap measure until we could do it using machines. And sometimes the data that comes out of doing it, you know, in stall catchers can then be used to train a machine to do it better, just as we used all 5 million stall catchers annotations to train GAIA, Laura's bot. So yeah, this is kind of the evolution of the process. So it looks like we've got about roughly a little over 10 minutes left. And Mike, did you have something else you were about to say?
Oh no! Just to emphasize the importance of the feedback that we get from the catchers, in terms of, I guess my role is to try to figure out how to deliver the best quality data and the best quality segmentation and all the rest of it, and that feedback is definitely helpful in that, and yeah, and things like innovations like that, looking at masking through citizen science as well. That, yeah, as you point out, anything that involves machine learning needs a ground truth, and in order to train machine learning, you need that citizen science side of it, or citizen science is a perfect way of providing that data that we need for the training. So it's an incremental process.
Yeah. Absolutely. And I think you just you may have just inadvertently labeled yourself the complaints department. So whenever anybody complains about the quality of a video, we're going to send them your way, because you're saying that's valuable feedback, right?
It is. Yeah.
Yeah. No, that's great. We really appreciate, and you know, Mike, probably everyone on here at one point or another for stall catchers has done something heroic, but I feel like, you know, Mike is often burning the midnight oil to get data stacks loaded into stall catchers, so we can get new data sets up and running quickly. You may have noticed that so you see Agla's name on here. Agla has been, you know, she was in the original launch meetings for the project. Anyone who's been with the project since the beginning knows Agla. She's kind of, I've focused more on the technical side, and Agla's done more of the community outreach. And of course, everyone knows Agla from her amazing blog posts, which has been such a great way to also catalog all the accomplishments of you know, the contributors to the project and the people involved. And I also want to acknowledge Libuja Vepshek, whose name you might see here. Okay, there she is. So Libby, as we're allowed to call her, joins us from Munich, where she's studying a combination of cultural anthropology and human computation. And Libby, so we're all doing our own research, and Libby's research is actually to study the rest of us. So Libby, I wonder if you could say a few words about what you've been doing over the past couple of years and why some people might know you from interviews.
Yeah, sure. Hello, everybody, and sorry for not introducing myself before. We had some issues that had to be resolved, but now I'm here, and it's nice to meet you all. So my research is about human-machine collaborations and human computation-based citizen science projects. So in projects like stall catchers, and I'm interested in the meanings that these projects have for the different parties, like participants and developers. And I've already had a chance to talk to some stall catcher participants, which was super interesting and also very impressive. And I've been playing the game myself and kind of observing what's going on at the Human Computation Institute, while also kind of contributing in some way or the other. So yeah, and this is actually also my first Catchathon, so I'm very excited and yeah, look forward to the next few hours.
Yeah, well, thanks a lot. We've really appreciated being under your microscope. I'm sorry, I mean having you as part of the team, and it's a you know, this new way of kind of coming together to you know, do research as a global community with scientists and non-scientists. Well, or in some sense, we're all scientists, just some doing it professionally and some doing it on a volunteer basis and some with certain expertise. And so you know, I'm actually very curious to read Libby's thesis when it's done, which is going to be some kind of a snapshot, I think, of how all these different parts fit together, all the different stakeholders and the culture around what we do and how we work together. This is really a new thing. And my sense is that this is going to be a growing thing. And I think, you know, COVID, you know, has created circumstances that have highlighted, you know, the extent to which we are becoming more of an online workforce, and you know, to some extent, how that's creating a kind of a gig economy, or you know, sort of amplifying that. And so I'm just very curious about how this evolution potentially impacts the future of labor in the world. So anyway, thank you, Libby. It's great to have you with us, and I know you're going to be joining us in Ithaca this summer for a few months to do some on-site work. So everyone at the Schaefer Nishimura Lab, you can run, but you can't hide. Libby's coming. There goes Mike.
Well, I would like to say, having been on the receiving end of one of Libby's interviews, that she does a just an excellent job. Yeah, she puts you at ease. She's very smart about how she goes about things, and being an old school guy who had no idea, if you ask me, well, how do you feel about, and I don't know how I feel about that, but Libby can get those kind of thoughts out. She's really fine to talk with.
Thank you, Mike.
Absolutely. I second that.
So Pietro, if I could just make a quick comment about where you're talking about the workforce, and I think anybody could be a catcher, right? Anybody can bring, this is all about empowerment to bring your own skill set to help advance research, right? And
right.
And we're talking about science now. We also want to talk about scientists and maybe the next generation and STEM education, like what you were saying, people with COVID, there's more online. My son himself, he's like, half the time now he's working from home on a computer. And I told you, Pietro, I sent this stall catchers to my son's school to let them know. But I mean, I think this is one thing that everybody can go and, use this to maybe get more kids interested in going into science. So I think with STEM as well. So
yeah, I agree. I think it's a great opportunity. I think it gives a new window on what science is and how it works and how fun it can be. And so, yeah, I hope, and I think a lot of us hope that this can be inspirational for students. I have to admit, I've sort of espoused a slightly different message when I visited all the students who participated and won that big team competition that we had when we launched the team feature. If you remember, some of you might remember that. 250 middle school students from Boise, Idaho at Hazelton Middle School. And I went out and met with them and awarded the first Citizen Science Trophy at the pep rally there. And one of the things I said to them is, you might think I'm here to tell you all to go and become scientists. Well, we don't want everybody to become a scientist. That could be problematic in a new way, but maybe some people would disagree. But I think part of the idea here is that everyone can understand science and be part of science without having to become a professional scientist. And I think having a population that has a greater awareness and appreciation of science means that the scientific enterprise in general gets more support from this, from these new generations. So I hope for those impacts as well. And I definitely agree with what you said, Diane. So it looks like we're just 3 minutes away from wrapping up the hour. I'm just looking at the leaderboard. We're certainly well underway. We're almost 4% in the first hour, through this data set. It looks like we've got a few small technical issues that have come up. I think Stall Catchers is much more performant than it's ever been. There were 100 catchers online this morning when I first connected to it, and it was humming like a singer. So we did some load testing with 800 players, and yeah, smooth sailing. So we, and I just want to thank a few members of the team. I particularly want to call out Libby and her partner Philip, who was also instrumental in setting up our load testing server. I just love how everybody pitches in and really appreciate all that. And then Ravish Dusaruth, who joined us fairly recently, I think within the last 6 months or so, as our new main developer. And Ravish, so Ravish lives in the middle of the Indian Ocean. I mean, not literally in the ocean, but almost in the island nation of Mauritius. And Ravish has just been very, very dedicated to making sure that we got the platform ready for this event, and spent long hours with me, working hard to leave no stone unturned, and the platform just wouldn't be running as smoothly as it is without his help. So just a really big special thanks to Ravish for that. And frankly, the whole team has really pulled it together for this event. It's one of the great things about doing a Catchathon is it brings people together in different ways. And I think it's brought our team together. Today it's bringing us together here in this Hangout, which I really am enjoying seeing you all and hearing from you. And of course, it's bringing the global community of catchers, including a lot of new catchers and a few schools and classrooms to do this. So many thanks to everyone. We're going to come back here. We're going to get our act together with the live stream, hopefully, for the next installment. We're going to do the final hour. It's a 24-hour event, so the final hour will begin at noon Eastern time tomorrow. We'll let folks know how to watch it on live stream. And we'll hope to hear from some of the teams who have been playing and see how the competition has heated up by then. And in particular, I'm interested to see how our robotic catcher has been doing. And it looks like someone just joined Unique Map. Oh, maybe we should say just a quick hello. Can you say a quick hello? Unique Mappers?
Hello, hello.
Hey.
Yeah, hello, good evening from Nigeria.
Yes. Yeah, one of our original international Catchathon teams. Welcome from Nigeria. Thank you for participating. Good to see you, Victor. How are you?
Thank you. Fine, fine, fine. Yeah. We've just sent our Eventbrite link for registration of our store catchers, and we look forward that within the next 24 hours, Unique Mappers will lead. Yeah.
Well, it's great to have you joining for this.
About it. Thank you very much.
Yeah, thanks a lot for participating. Wouldn't be the same without your team joining. So yeah, nice to see you again. And we're wrapping up the hour, so that's the perfect way to cap it off. Good luck to everyone, and we'll see you in 24 hours.
Okay.